Are there any questions about the material that has been covered till now? And Srijan already has a question. I'm going to cover that. Are there any additional questions? Jacob? Do you have any questions? Sure.
Maybe quiz them, ask them a few questions.
Okay. I'll ask them. I'll ask them. And so
What's the attendance sheet? The attendance sheet?
it's in my mind.
I'm having it, all of it. On the
I remember
computer.
it.
No, it's there. If you go and see participant list, it says bye. I just did it.
Yeah, I know.
Do you have 100%?
Okay, I'm going to go back to dihybrid cross, and I think Srijan asked the question. There were 9 genotypes, right? Does anybody remember 9 genotypes? Yes, Srijan. Your question was, why is it 9? How can you figure it out in a better way, right? So we are going to do that, but somebody is going to help me construct a Punnett square first for a dihybrid cross. I know you can do it. I would like somebody else who wants to volunteer. So maybe this is going back to what Mendel did, a tall and a short. So tall, and then dihybrid means there are 2 alleles, 2 genes, or 2 traits, and tall and round. So this is the starting point, and you want to come up with a Punnett square. Sharon, do you want to do it? Do you want to come up with a Punnett square for? No? Nandini, you want to try? Amy?
I want to try.
Amy, you want to try? Brandon, you want to try?
Yeah, Brandon.
It's tall and round. It's a dihybrid cross. A tall and a round with a short and wrinkled, right? Assume that round is dominant over wrinkled. I don't know if that is. Sorry? Yeah, but remember the notation is the dominant one is capital. Whatever is the recessive gets small. It may not be, and so usually you actually use the letter for the dominant as the capital letter. That's the notation that people usually use. And Amy, you want to try? Why don't you give it a try? So tell me what happens in the first generation, and then second generation, draw the Punnett square for the second generation. And then you have 16 of these, right? Why don't you fill up the 16 of these, because we have to count and make sure that we get 9, and then I'll come to the formula, like how you get to the formula, because it's pretty easy. Does everybody get this? What Amy did? Any questions? Anything wrong she did? Okay. So now the question was, how many different genotypes do we have in this?
We have so many.
And what's the
We have so many.
how many different genotypes do we have in this square? How do you identify the different genotypes? How do you identify which is a different genotype in this square? Yes, Shobhadru?
You see which one repeats and those like, so if it's a different genotype, then it doesn't like repeat basically.
It's not quite true, right? It's not about repetition. It's, so in this, when will you say that a genotype is the same and when will you say it's different? Okay, Anushri?
Same genotype is something that was from the original parents. The different one is something that's not like.
No. Go back to what does a genotype mean? Yes, Srijan.
The genotype is basically the genetic
Makeup.
material. Yeah,
Okay, so in this thing, in this square, how will you tell which ones are the same genotype and which ones are different? Like, how do we come up with that number 9?
Well, first of all, on the left side of it, those 4.
Mm-hmm. No, the genotype, first of all, what is the genotype in here? This is one genotype, right? This is another genotype. How can you say that these are different?
Because the coding is different.
Yes. So, for example, this one, this genotype and this genotype, even though they came from a different combination, they are exactly the same, right? You see?
I kind of switch that one up on just that one.
Sorry?
It means the same thing.
Sorry, I didn't get that. Yeah, so exactly, exactly right. So what you did here is you took from the, let's say this is the father and this is the mother. So you took something from here and you mixed it up with something from here and actually this is, sorry, the top. This one with this one is this one, and this one with this one is this one, right?
No,
Is there a mistake here? I think there is a mistake here.
First one on the top.
Actually, this should have been,
No,
oh, okay. It's just written in a different way. So, this one and this one, so these two genotypes are exactly the same, right? Even though they are coming from different combination of the parents. So the way you identify it is this is the genotype, right? So if you look, this is one genotype, this is another, let's just mark these. This is one genotype, this is another genotype. Now this TR, this is another genotype, number 3. Now are there other TRs? Yeah, right here. So this is number 3. And so on, right? You can mark them. Right?
No, the one on the bottom is
It's
lowercase
different.
t.
It's different? Oh, yeah, you're right. But it must have another number 3, TR. No, actually, this won't. This is unique. This will be unique. And I'll come to why these are unique. But number 4 would be TR, number 4. And is there another TR? Let me just go back. So this is this TR and this is this TR. So there has to be another combination which is this TR and this TR, right? Which is this. So yeah, this TR. This is also number 4, right? These two are the same. Everybody see this? Because it doesn't matter whether you write it in this order or this order. It contains exactly the same content, right? So you can identify this, right? So now the question is, how do you find the total number of genotypes that will be there in this kind of a problem, right? So another way of representing this would be, this is really a problem of combinations, right? So another way of looking at it is, you have ER, this is 1, you have TR, this is number 2, then you have TR, this is number 3. Actually, you shouldn't go that way. Let's start with this, right? In the first generation, what are the things you will get? You can get a TR, right? That's your basic unit. So let's see. So combination 1 is TR, and TR, right? And you are combining this with another TR, and TR, right? So how many different ways can the first one combine? This is unique. This is unique. Now, this and this, let's see, TRR, this is unique. TRRR and TRR and they are all unique. There should be 3 of these, right? I'm trying to remember now. Oh, okay. They are all unique, but now let's do the other ones, and let's see what happens. Now, when I do this TR with this, for example, I need to. Okay, let me back off. Let me do it another way. I think this way is, this works, but it's again this complicated. So let's say we didn't have the, second attribute, okay? So if you just work with TT with TT, right? Because that's your first generation. What do you get? You get a TT, you get a TT, right? And then you get a TT, and then you get a TT, right? Suppose the R wasn't there. Now, how many of this do you have? You have one of this, right? And you have one of this. And then, how many do you have here? You have 2 of this, right? Now this is getting combined independently with RR, which is 1, and then again the same thing, R, R and RR, which is 2, and then RR, which is 3, right? These three are completely distinct, and these three are completely distinct. And how many ways can you combine them? You can combine this with this, this with this, this with this, right? This with this, this with this, this with this, and then this with this, this with this, this with this. And so, the number of unique combinations you can get is 3 to the power of 2, right? Or 3 times 3. So that is really the formula. And if you counted here and looked at this picture and tried to identify, you will get exactly 9 different combinations. Does that answer your question, Srijan? Okay, now the other question is, so this is the number of genotypes you have. Now, how many phenotypes will you have, given that one is dominant and the other is recessive? How many would you have? Guess. What would be the formula for that?
Is there 9 different genotypes?
What's the question?
In here, you have 9 genotypes, right? And we kind of came up with how we have 9 genotypes, because you can just look at the characteristics independently and then combine them. This is the law of independence. And if there are 9 genotypes, will there also be 9 phenotypes? No. Will it be more or less?
There would be less.
Less,
Always
right?
less.
Good. So, question is how much less? And how do you get that? Can anybody figure out how to get that? Can anybody see? I mean, basically follow the same procedure that we did to get the genotypes, to get the phenotypes. Yeah,
I'm guessing 3 then.
3 phenotypes. How did you come to 3?
I'll guess 4 because it should just be tall, round, tall.
Absolutely right. So, exactly right. So just think about it. Here, how many phenotypes can you have? Only 2, right? It's all binary here because it's dominant and recessive. So 2 here and 2 here. So 2 square. And 2 square phenotypes. Does everybody get that? Right? And any questions? Okay, now tell me, if these 2 characteristics were incompletely dominant, how many genotypes and how many phenotypes would we have? Or co-dominant? Does anybody remember what incomplete dominance meant? What was it?
Me?
Anybody, just yeah.
Let someone with recessive,
Sorry.
It's like you let some of the recessive,
Yes. Always remember the pink flower. The red flower and the white flower became the pink flower, so they get mixed up. And what is codominance?
Stripes.
Yes?
So like, there's like still some of the recessive shows up, but like it's separate. Like the colors aren't blended. There's
Okay, good. So now if this was, these two characteristics were incompletely dominant, which means, what would, in this case, incomplete dominance mean? Tall and T and T would lead to what kind of a tree?
A medium plant.
A medium plant, right? And if it was, well, codominant doesn't make sense in this case, but, okay. Then, with incomplete dominance, how many genotypes would you have and how many phenotypes would you have? Just look at this. The answer is there in this picture. You just have to interpret it differently. How many genotypes are there in this row if it's incompletely dominant?
There are 3.
There are still 3, right? This is a long plant, this is TT,
Oh, 3
TT and
times
TT,
3
right?
equals
9. So
9.
that's
Yeah,
so it's the
It's
same.
still the same.
Genotype is still the same,
the
phenotype
phenotype
is what?
I just said to you.
Is
I
9.
told you.
Is 9, exactly, because now this becomes,
Not necessarily.
Distinct.
Wait, I got this.
earlier, this, because of the fact that one was dominating, you couldn't see the difference between these two, so you had to club them together. Right? Is it clear? Now I'll give you a more challenging question. So there are 16 different combinations here, right? 16 different combinations. And they are mapping into 9 genotypes, right? And the 9 genotypes are what? You can figure out from here, TTRR, TTRR, TTRR. You can just write them down, right?
Yes.
And these 16 are mapping into the 9, right? There are 16 of these and they are mapping into 9. So bigger number mapping into smaller number. What would be the ratio? What would be, so there will be like 9 different numbers, right? And they all have to add up to 16. How would you approach that? By the way, this is something that they ask you in Science Olympiad, yeah.
Can you repeat the question?
Okay, so there are 16 different combinations you can get from the Punnett square, right? 9 of them are unique, right? So how would you map, what is the ratio of the 9 of these such that it's 16? Yes, Amy?
My mom told me to remember it was like 1, 1, 2, 2, 4, 3, 2, 1, 1.
Very good, but why?
Because I don't know how to explain it.
Because there's
It's there in this picture.
because there's 16 of them,
No.
and then because of the probability of each one, because in the Punnett square is suppose like a capital T, capital R, capital T, but lowercase r, it appears 2 times.
Mm-hmm.
And then compared to that, like others
Mhm.
appear even more times. So
Mhm.
1, 2, 3, 4, blah, blank, blank, blank.
Excellent, excellent. That is the answer, but did everybody get what Srijan is telling? No. Okay.
No.
Okay, okay, so we have these 2 pictures, right? These 2 pictures people understand, right? Okay, now I'm going to write this, the different genotypes now in this way, right? There is a TTRR, then there is TTRR, that's this with this, and then there is TTRR, right? And then let's write the next one. There is a TTRR. Then there is a TTRR, and then there is a PTRR, right? And then there is a TT RR, there is a TT RR, and there is a TT RR. I made a mistake, this can't be a TT RR. This has to be a
All of them are
TT
TPRR.
RR.
Yeah.
All of them are TTRR, yes. But note note which one is capital and which one is small. Okay, these are the 9 genotypes. How did I get it? I took this combined with these. Those are the first 3. And then the question was, how many of these are there? So there are only 9 here, right? But they have to add up to 16, because that's what is there in the Punnett square, that's the total number of combinations. And so what Srijan was trying to tell was, if you look here, there is one of this, and there is one of this, right? So how many combination of this will be there? There will be 1 times 1. So TTRR will be 1. Now look at this combination. There are 2 of these, and they are combined with 1 of this, so there will be 2 times 1 combinations of this, and then 2 times 1 combination of this, right? So that's where the 2 comes from, right? So wherever is the TT, RR and RR, there will be 2 of these, right? And then this one, there will again be 1 of this and 1 of this. So this TTRR, there will be a 1, and the TTRR, there will be 1. This TTRR will be 1, right? Everybody see that? This one with this, but there will be 2 of these, right? The small TRR, there will be 2 of these, right? And then this one, how many will be there? 4, because there are 2 and 2, right? So
See?
this one, this will be 4. So then the ratio will be 1. So now you can write the ratio. There will be 9 numbers to write. You can say 1 is to 2 is to 2 is to 4 is to 2 is to 2 is to 1. 1, 2, 3, 4, 5, 6, 7. No, I think there will be. There is another 1 and another 1. So 1 to 2 to 2 to 2 to 4 to 2 to
1 to 2.
But do you understand why your mom told this to you? Does it make sense? By the way,
It might also be easy for them to figure it out directly from the Punnett square, right?
yes,
So you can check, for example, uppercase both, TR, only 1.
only 1.
Then you write 1 on the side.
Yes.
That's the way I learned. And then you say PR capital, T smaller, how many and then you write next.
That's
So
it.
visually you can look at it.
Visually you can look at it.
And then just write it, so plus like you do.
Yeah, but the thing is, here you have to count all the 16 and you have to figure out which ones are the same, and then you have to.
If you draw the Punnett square, then
That's right. Yeah, but here you can basically figure it out just from, like, just looking at these
maybe you
combinations.
can mention that to them so they understand how this and that is related.
Sure. Huh?
That's a 1, right?
This one?
No, up there. No, down. At the ratio.
This is a 1.
No, after that.
4,
1
4,
to
4,
1
4,
to 1 to 1 to 2 to 4. The 4 comes because of this, and you can see in the Punnett square, this is occurring 4 times. Look at this pattern, T, the capital T, small t, capital R, small r. Let's look for that pattern. Capital T, small t, this one, this pattern should be occurring 4 times. Just see. Capital T, small. Where is the next one?
Oh, you're right.
This one is the same. Okay, let me mark those.
No, no, the
No, not
next
that
one,
one.
the next one.
Next
Because
one.
that is too capital R.
Next
The
one.
next one.
Next one, yeah. That one.
Yeah, also the one below the one.
This one,
Also the.
And the one below that.
the one below that?
No,
No,
the one on the other side. The one on the.
No, this is also not. Yeah.
Lower, lower, lower. Left, left.
Third column, second row.
Where is the next one?
Third column, second row.
Underneath the one you made a mistake on.
Here?
Underneath the one you made a mistake on.
Just diagonally up.
Yeah. Next to that.
No, left.
Left.
Yes, here.
Get that? Maybe you can ask them to go.
Yeah,
Why
specification.
don't you pick up?
At least that they can do, right?
Why don't you pick up and make sure that this actually works?
And then the bottom left.
Ask them to go.
And then the
bottom
Brandon,
First.
find the fourth one.
first column, first
I know.
This
Okay,
way.
and you will do the next one.
The fourth one is right here.
Oh,
Yes.
It's just like a staircase.
Yes.
Naomi,
It's diagonal, very nice.
you give me
Yeah, it's a diagonal.
from, actually I'll give you another. You give me the other two.
It's like a game.
Give me this combination. T, R,
Why
R.
don't you call everyone? One by one.
Give me this.
Go, two capital T's and
Two capital T's.
one capital R.
One capital R, one small
Figure
r.
it out, just look at both. Two capital T, look for that.
Oh, yes.
And you can take help from your friends if you want.
The girl next to you. Anushya, hello, sir.
Where is the other one?
God, it's exactly the same.
Excellent.
To the
Thank you.
manager, how we doing?
How about this one? Capital T, small t, small r, small. Jacob.
Baby.
Get up there,
Go,
Jacob.
go, go.
This one. Capital T, capital T, this capital
All you
T,
have to do
small
is box
t,
it. Ricky
small r, small r. Okay, you use the green.
Come on, you have to do it. It's like
This and this.
find the hidden. Always diagonal.
Ricky can't do this, Jacob.
Come on, Ricky. Whoever Ricky is.
Dude, that's Jacob, that's Jacob.
Wait, so that's Ricky?
No, that guy's name is Jacob.
Who's Ricky?
Probably Ricky's this other guy. Okay, then are you talking about Ricky?
Okay.
English or validity repeat.
Are you mentioning this in heredity?
Capital T, small t.
Ricky related?
यहाँ पे किधर गए नहीं
Yes,
तो
that's
कोई
one.
Yes,
Here's the other one.
yes, yes.
Great.
Blink.
So now you can see, right, you can look at these Punnett square and you can see what I did here. And does it make sense? And you can come up with this ratio. When you come up with the ratio in the science Olympiad, they give you a dihybrid cross and they ask you to give a ratio. If you do it by counting, it will take you a long time. If you do it this way, it will be shorter. Right? So this is the trick to use. But you should not have to memorize anything. All you need to know is the rules and then apply the rules and it will work. It will just be like this. Does everybody get this? Okay, good. Srijan, is your question answered? Excellent. Now, I'm going to tax your brains a little bit more. Okay. Suppose you were to do a trihybrid cross. And that's not there up to the state level, but when you go to the nationals, you will have to do a trihybrid cross. Okay? And I'm expecting some of you may go to the nationals, right? But anyway, that's not the point. Based on what you just learned. Okay, what would a tri-hybrid cross look like? Tall, around, and let's say a white, right?
All you have to do is do it.
With
It's really easy.
short, wrinkled, and let's say a purple.
Hey, quiet, quiet.
White. First tell me, how big is the Punnett square going to be? You don't have to draw it because it's going to take a while. But how many will be there? What would the Punnett square shape look like? How many squares will be there?
Yeah, 6.
All right. Tell me.
Wait, no, there's going to be,
9 by 9?
wait, so T.
Why 9?
Wait, no.
I should also do this.
6 by 6?
No.
There's 3 of each of them, and in the last one, there's 2 of each of them,
Yeah,
and
6 by
4 by 4, and so now you square it, yeah, 9 by 9.
I think it's 36.
9 by 9?
Yes.
Who said yes? Yes? 6 by 6? Srijan?
64 squares, 8 by 8.
8 by 8. Excellent. And why 8 by 8?
Because in a monohybrid, it's basically 4 to the how many genes there are. 4 to the third power in this case. So 4 times 4
4 times 4 times 4.
equals 64.
64, right? So how many rows will be there? How many columns will be there? 8 and 8. Very good. There will be 8 here and 8 here. And the reason there will be 8 is so go back to the basics. So any individual will now have these 6 things, right? Right? And you can pick the first one in 2 ways. So there will be, so one way will be T R W. You can, that is one combination. Then you can pick T R W. So basically you can pick the first one in 2 ways, the second one in 2 ways, and the third one in 2 ways, right? So what is that? 2 times 2 times 2. So that's 8. So that's why there will be 8 here, and there will be 8 here. And if you have 8 and 8, if you multiply that, that's 64, right? Now tell me, how many genotypes will be there in the 64? You did it for 16. And this, if you have understood the 16, you don't have to count and tell me, but tell me how many genotypes will be there. Brandon? Just think.
Okay.
Is that 27?
27, yeah, that's what I said.
27.
That's 27, I think.
And why is it 27? Yes?
Because it's what you said, the T, it's the same diagram except there's another like W being added to the bottom, so it's
Exactly.
like W.
So that means 3 times 3 times 3.
3 times 3 times 3. Exactly.
Oh, yeah.
Exactly.
cute.
And how many phenotypes will be there?
Phenotypes 3.
So then for phenotypes, there's 8, so
8.
2, 3, 2. Yeah.
You get it?
Yeah, 3. Right.
27 genotypes, 8 phenotypes. Can you do the ratio now?
Yeah.
The ratio of wait, which one?
All the different. So now 27 will go to 64, right?
Okay, so.
And if you try to do it in this way, it will be very hard. But you can do it in this way. See, all you have done is you have added something here. So now if you look, you'll see same thing. You have WW, you have 2 of W, W and W, W. And then you have two small w's, right? So what are, and you just keep drawing. So 1 times 1 times 1, that will be one combination. 1 times 2 times 2, so
Oh, so
that's another
27.
4. Then again,
Yeah,
this is 2.
It's the same thing.
So there'll be a 1,
It's the same.
there'll be a
It's the
2,
same thing,
there'll
obviously.
be a 4,
Yeah.
It's 27.
and then there'll be a 8, right?
It's always the same thing.
And then you just have to do this exactly like the way you did the 4
The
by
first,
4.
yeah, the first.
Yeah, so.
Yeah.
Can you move the screen back up so?
Yeah,
Oh.
because it's like
It's kind of
Noise.
So without going into the, without actually drawing a Punnett square, when you do problems, if you think of it like this, it'll be much easier for you to do the problems because it's nothing but it's a problem of combinations, right? And as long as you remember the basic rules, it's pretty straightforward. By the way, in Science Olympiad, these kind of problems are there. So, and you'll only get better by doing practice. I mean, you know the stuff. There's not much else you learn, but unless you practice, you'll not get speed, and that's, I guess, one of the important things you have to pick up if you have to do very well.
Is there any sample question paper anywhere? Like what
Yeah,
they
actually, I
can practice?
uploaded some. I uploaded some sample questions for trihybrid cross. No, and I'm not going to ask. My intention was not to actually give any problems on that, but at least I wanted to see how much they have learned, and looks like it's not hard, right? It's pretty easy stuff.
Quad hybrid.
Just remember.
That was 16 times 16.
It's going into maths now.
That's 16 times 16.
Maths and
So what I did was trihybrid cross. I'm not going to spend any more time, but you should be able to get the ratios, right? And just one last question. In the trihybrid cross, how many phenotypes will be there?
You said 8.
You already said. If there was incomplete dominance, how many phenotypes will be there? Excellent. That'll be 27 because genotype and phenotype become the same because this one, the reason why this was 2 is because these two counted as 1, right? Because this guy was dominating, this little t didn't matter, it was dominating. So as Amy had said, it's really there are only two kinds here. There is a tall and a short. There is a round and a wrinkle. There is a white and a purple. So 2 to the power 3 is 8. But if this happens to be incompletely dominant, this will show up as a separate phenotype, right? And so genotype becomes same as phenotype in incomplete dominance or codominance. Okay? Okay. So I'll now cover So what I'll do today is cover a few things that we left out last time, what is there in Science Olympiad, in your syllabus. And what were we covering? We covered 3 things, right? As part of exceptions to Mendel's law, we covered 3 things. What were the 3 things that we covered? Yes?
Incomplete dominance.
Yes?
Codominance.
Yes, and then what was the third thing?
Is it multiple?
Oh, multiple
Multiple alleles, exactly,
Multiple alleles.
Oh.
exactly.
That's why I forgot what it was called.
And the reason they are exceptions to Mendel's law is because of what?
Because what he tested, it was on pea plants, and that wasn't gonna. It might work on some.
No,
characters.
remember his laws. His laws said that there are only two traits, two different possible traits, two variations.
Oh yeah.
With multiple alleles, there are three variations,
Even
right?
more sometimes. Yeah.
And then with incomplete dominance, it's not that there is one dominant and another recessive. There is now, if you mix them up together, you get something else, right? So it's not following his laws, right? And it turns out that there are, in fact, Mendel's laws were the exceptions, because there are so many combinations that can happen, which is why people didn't notice before Mendel what Mendel noticed, because usually when you have like two parents, the child doesn't resemble like some characteristic of either parent completely, right? There is always some blending, they're different, right? And so this is why people didn't quite get what Mendel was saying, didn't quite understand. But Mendel actually, the real value that Mendel brought was the fact that he made people think in terms of these basic units, which later on, I mean, people as they researched more, they figured out what the actual reason for this was. And that I'm going to cover that later. But before going there, I want to cover the other exceptions that exist to Mendel's laws. Let me try. Okay. Oh, okay. There you go. So, see, everything is here in the class presentations folder. You will see there's a lesson handout where I've kind of summarized things. Yeah, I just put this into one thing. So this is your syllabus. This is taken directly from the Science Olympiad sheet, and as you see, these are the topics that we are covering, and we covered up to here, right? And I also covered sort of this part. What today I'm going to cover is this epistasis and multifactorial traits.
Oh god.
Okay. Now these are yeah.
What about cell introduction, molecular?
Those will happen. Yeah, I'll try to see if there is time. I'm going to kind of give an overview of. It'll be like the trailer of the rest of the movie. Okay. And then.
Harry Potter 7 part 2.
How much time do we have?
Yeah. 30 minutes.
30 minutes.
20.
It's 7 minutes.
20.
3 minutes. 3 minutes.
Multiple alleles. This we actually covered already. In terms of what? Does anybody remember? What was the example? What?
Blood groups.
Blood groups. How many alleles were there?
There were. Oh god, there's 1, 2, 3, 4, 5.
How many alleles were there?
Alleles is 1, 2, 3, 4, 5.
No, alleles.
Hey,
There were 3 alleles, right? There was an A, there was a B, and there was nothing, right? I. Alleles, remember, alleles are the different versions of of what can happen.
Those are the
When they combine, they can give more combinations. That's what the genotype is. That's what the phenotype is. Alleles is the basic traits, right? So here and there, what was the other interesting thing about this blood group? Does anybody remember? Yeah, Srija?
The A and B,
Mm-hmm.
there were two possibilities for
And
the blood.
what was that thing?
One was IA, the other one was IA.
And what was that
loci.
characteristic?
The characteristic was
What was the characteristic of the A and the B alleles? They were co-dominant, right? Yes. So it was multiple alleles and co-dominant. Now look at this. This is the 4 kinds of rabbits you can have. And here, can anybody tell me what are co-dominant here? Is there any co-dominance here in this?
no.
Is there any incomplete dominance here? What would happen if these two mixed up?
There would be something in the middle.
That would be like
Because both of them are co-dominant.
Yeah, and so the reason I'm showing you this picture is because this is an illustration of 2 things. One is multiple alleles, not 3. There are 4 alleles here. Do you get it? Get it? And then there are rules by which they mix, right? Because if both of these mix, what is going to happen? Some kind of incomplete dominance will happen because
Will
both
be a
of them are
mix of them.
recessive. If there is a full coat color and it mixes with any of these, then you will get a full coat color because this is dominant. But then if these recessive guys mix, then you will get something else, right?
No mix.
You will get a mix. And you can again do the Punnett square and you can follow the rules and you can figure out what the colors of the rabbits are going to be. Yes?
The Himalayan one was like mixed with the albino one, right? Because they're both white with red eyes. And, well,
This one with this one?
Yeah, like this one was mixed, like the D was
Mhm.
mixed with something else and that
Yes.
made the Himalayan one, right?
That's exactly right. That is exactly right. This is purebred and this is purebred because it's got a full coat color and this is the albino. These are all mixes. How many phenotypes are here?
There are 4.
Very good. How many alleles are here? Excellent.
Yeah, 4. 4.
Does everybody get that?
Yeah.
How many genotypes will you get?
4.
4?
There are 4 different.
Well, listed are 4 different allele genotypes.
How many genotypes can you get out of this combination, out of this, given this?
16.
16?
Since these are all alleles,
Yes?
genes would be
16.
16.
Does everybody get why there will be 16?
yeah 4 times 4.
Each rabbit
Because
so
each
you can make
has
Punnett
2
square
of
with the different rabbits.
Exactly.
That's
Exactly.
4.
16.
16. Good job.
Get it?
Just do it times
Now, this is interesting, right? With one gene, that's a monohybrid cross. Remember in our earlier pictures? And how does a monohybrid cross Punnett square look like? How many rows and how
2
many
by 2.
columns?
2
2
by
by
2.
2, right? One gene, right? One trait. But because now there are 4, you have a Punnett square which has
4 by 4.
16 squares and each will have a different genotype, right?
Right.
And what about the phenotypes? Do you think all the 16 genotypes will result in different phenotypes?
No.
No. Why?
4.
Because 9 of them will be.
9?
Wait.
Not 9.
The question one more
You have to
time, please?
think.
Wait, what was the question?
Question is, there are 16 genotypes here, right?
Mhm.
16 distinct genotypes. Will that result in how many phenotypes?
How many phenotypes? No, 3. No. Mm. Oh, the ones. Okay. So this is a hybrid. Yeah. Should be 9.
You see 9? Okay, let's.
16 genotypes.
Okay, actually there is. Okay, this is also very interesting.
Huh?
There is a hierarchy here. So there's rule. This is dominant, but among these, this is dominant over this, is dominant over this. This is interesting, right?
Oh. Okay. That is weird.
That is weird, right?
Makes sense.
So that is exactly what I want to tell you. That there are so many exceptions to Mendel's laws that it is weird. It's multiple
Oh, wait.
allele.
Isn't it because this middle two are basically combinations of C and the two because of those? Because then they already combinations of.
No, this is just among the different alleles. If you have two alleles together, you'll always have two versions of a gene together, right? In any individual, you know, TT, you have to write 2, right? So if you have this and this, then this will dominate over this. But if you have this and this, then this will dominate over this. Now, if you have this and this, then this will dominate over this. Right?
Right.
So let me give this as a problem to you, and I'll type it up and send it in email. Why don't you find out the number of genotypes, which you actually already did, and then
16.
how many phenotypes are possible?
4 phenotypes.
4 phenotypes?
Yeah, because
Oh yeah.
dominance.
That's true.
There are only 14 phenotypes. Because
And they're all the same. We solved it with the moment we were given it, which was like
2 seconds.
it was like 5 minutes.
2
It
seconds.
gave us the phenotype.
Oh well, whatever. We weren't given all the information.
It was pretty obvious that the dominant names were dominant.
Oh yeah, you also have to think about
Actually, so you see, there are not 4 phenotypes here. There are 5 phenotypes. Okay, so the reason I'm showing you this picture is because in this case, it's not just, so this is what is the, some of this are temperature sensitive, so this is also important to know that the genes, how they get expressed, how the allele gets expressed, sometimes depends on the environment. Okay? So that's another factor which you should be aware of. It's complicated. Biology, there are so many different variations going on. It is complicated. Yes?
Is it like if you have brown hair and then if you stay in the sun a lot or something, your hair turns like blonde or something.
Yeah.
Because that's what's happening to my friend.
Oh, what? That's interesting.
I don't know if that is because of some genetics or is it something else.
That's just a chemical reaction.
Okay. But if he, so the way to look at it is, if he goes back, wait, hang on. No more talking. You got to listen. If your friend goes back, the way to test this out, how would you test that whether this is actually happening because of the environment, his genes have changed, or it's something else? How would you test that out?
I'd make her stay in the sun a lot and see if actually
Cut off
And
some
then
hair.
if she went back home and just stayed at home, what would happen?
I would see if it was turned back to brown.
It'll turn back to brown.
Yeah.
Okay.
That's gonna be hard to do actually.
But
This
they'd
is
have
going to be hard to
stay-
do.
they'd have to
That's
stay-
actually not gonna
they'd
give you
have to stay in
correct
the
results.
classroom for
Not
like
gonna
a
give you
whole
correct
year.
results.
Because
They'd have to stay inside for like a whole year.
Yeah.
because there are also chances
It
that
might not, like, if it changes once, it probably won't change back.
That is true, and that is exactly what happens in the cat. If when the cat is just growing up in the womb, if the temperature is different, it will turn out to be one way, and that's how it will be for the rest of the life. Anyway, this is something that I somewhat covered, but I want to cover again. Lethal alleles. Lethal alleles means it
If,
is lethal.
yeah.
Can anybody say, can you have a dominant lethal allele?
No.
Why not?
Because if both are
You're
dominant,
already dead.
you're already
Exactly.
dead. You're already dead.
So lethal alleles are always, yeah.
Sorry, I have to leave right now.
Okay. I'll be done in like a few minutes. And you can watch the video afterwards. Yeah. So lethal alleles are always recessive. And one of the things that you'll notice is because a version where both lethal alleles are present actually doesn't survive, in reality you will never see the ratio of the genotypes that we saw, the 1 to 2 to 1, you'll never see that. You'll only see a 1 to 2. Right? Because the third one is gone. It doesn't survive. Okay, and there are some, and this is some temperature sensitive lethal alleles. So again, the point is environment plays a factor in how your alleles get expressed. Okay, I'm going to skip this right now.
Wait, what was number? What was last slide? Can I see it?
13.
What is, oh, it's a chicken.
Yeah,
What about 13?
13.
13.
13.
Yeah, 13.
That's a sickle blood cell, I think.
Yeah.
Yeah.
Wait, sickle cell anemia? Oh.
Yeah.
So what happened?
What happened?
Wait, what happens in a sickle cell?
Where are we all going?
Okay.
It's like hash. It's like web. Because I don't want to be a doctor.
Actually, why don't I, because I haven't read this up very well. If you're interested, I can cover this next time. But
Yes.
Yeah.
I just want to show you all the different relations that are possible. And this is, so again here, this is what I was trying to show you. This recessive allele, one of that is dead, right? And the reason it survives, that allele at all survives, is because it's recessive. And if it's recessive, it's like hidden and it shows up But it doesn't really show up because before it can show up, it's dead.
Creeper, normal.
This is an example of
What does creeper mean?
Yeah, what is
creeper. I think it's
can hatch
it's
dead.
Completely
the,
deformed.
the short-legged chicken. Creeper means its legs are shorter.
Oh.
Okay.
Oh, is that why?
And yellow mouse and the normal mouse. Yellow mouse is always lethal. So if you have 2 copies of the yellow allele, you are dead.
I have a little question about the lethal alleles. Has there ever been in a lethal allele, has there ever been like a codominant gene that has a some unknown characteristic and a lethal allele at the same time?
Yeah, actually, I don't know. Maybe one of the other parents know if that happens.
Probably not, we'll have part of the tissue dead, that means basically that is not gonna happen.
That's
But-
gotta be half dead.
But but one thing that can definitely happen is, may I have your attention, please? One thing that can happen is some of the lethal alleles can get turned on at a later point in your life. That definitely can happen. So you will survive, but you will die.
Eventually.
Eventually, everybody will die, but you will die of a particular disease
Caused
pretty
by
much
the lethal
in your, either as a child or as a young man, and that definitely happens. Yes.
So some diseases are like based on that.
One disease is this Huntington's disease, and I'll try to read up more and kind of, I think it's called Huntington's disease. Okay, and then and this is something that I finally want to go over. This is called epistasis, and in epistasis, one particular gene starts impacting the effect of another gene. So suppose one version of an allele is there in one gene, another gene will appear differently. This is again against Mendel's laws because it's not independent, right? What Mendel said was every trait or characteristic was independent of each other. Here one is affecting the other, right? So that is called epistasis. And I think I'll kind of go over this. There are a lot of nice pictures, and then this is another. This is called polygenic pattern, where multiple genes actually result in one color, like one characteristic.
That means like it's incomplete dominance within 3 of them?
It's not quite incomplete dominance because each of these genes don't really express on their own. They don't express anything on their own. As a combination, they express something. So you can think of these 3 genes as almost like effectively one gene. So again, these are all exceptions to Mendel's laws, and I mean anyway, height is one polygenic inheritance. The height is determined by multiple genes, which is why you have many different kinds of heights in the population. Okay, this is pretty much, so what I'll do is in the next part of the next lesson, I'll go over these examples in some detail so that the concept gets in.
No, down. No, it's not the chicken. Yeah, no.
Chicken?
No, I want to see the epistatus disease, like, the
Definition.
Yeah. It's
Okay, you can turn the lights on and
Type them up and use it.
and one of the things in heredity that you have to do is you have to remember a bunch of different terms. This polygenic thing, pleiotropic, epistasis, you have to remember what they mean and apply it when the question comes. But going forward, after I cover this I'll probably spend half of next lesson covering this. Going forward, till now what we have been doing is we have been saying this is the rules, this is how you kind of apply the rules and then solve these problems, right? What we have not done is we have not done why this happens, right? What's the basis of this? And it turns out that while Mendel and others did this just by observations, they didn't really know about genes. Subsequently when scientists actually went at the more molecular level and saw what was going on inside a cell, they actually could figure out what was going on which could cause both Mendel's laws to work and also the exceptions to Mendel's laws to work. And then the other question that all of you may have is, why are we different from our parents? Right? Because if you just followed Mendel's laws, you should have had something from each of your parents, right? But that's not the case. We are completely different. And as you go into how cells divide and they come together to form a new life or a new cell, you will see why this happens. And the reason also is, in Science Olympiad you'll be quizzed about it. So that's why after this set of slides, I'm going to go into cell and cell division and the different ways cell divides, and then genes and chromosomes, how they relate, and then how the fact that you have these different patterns eventually lead to different people or animals in the same species. That's what I'm going to go over for the rest of the course. And that should cover most of at least the topics that are listed in Science Olympiad. I think I will take probably another 5 lessons to cover all the topics. So that will be next one, and then 4 others. All of you there, the day before Thanksgiving, the break, that's a break for you, right? The school break. So will we get that room? Will we get this room?
No.
Okay, then I'm not sure. Then I think
whole week, like a lot.
you're off. So what you can do is,
We could do
maybe
it
that
at someone's home,
well, but doing a lesson won't be very easy. We can do problems maybe.
you could meet and revise.
We can meet and revise and do problems.
But
I'm
we
fine
won't
with
be
that.
there, I won't be there.
And then I'll have.
But
how
then
many-
only 2 more weeks you're there.
2 more weeks before I
going
go
to-
on vacation during the Christmas break.
So basically then 3 weeks, next week,
Yeah.
and then 2 more December. But you're saying you need 2 more, 5. So
Yeah.
then
So I think
January will be,
Either it will go on January or
Or
Simpson.
or if Simpson covers.
While I'm not there, that will work.
21st week. So we need to revise,
Actually, it won't be available.
Where are you going for Christmas?
I think we can still cover it in January. We should be done by the middle of January if we just took the breaks. And one thing we could do is during the breaks, I mean, I can work with the other coaches and come up with some problems. And then maybe people can solve those problems, because that also will take time. And then we can resume. So at least then you're very confident about the stuff that we covered. And then maybe we can cover the rest of the topics later. Yeah.
We have like a Google chat and then
I'm not sure how it will work. We have tried that before and
How about a blog?
blog? No, I think we'll end up.
I saw one
Google
topic,
Hangout.
and then we create comments and questions on top of that.
So how about this? In the
Emails,
days in which
emails,
the school is not going to be there, Zoom is not available, we just come up with problems and assign problems to people for.
Can you maybe email out the dates specifically? So that, then I can see my schedule and that will help me to.
So really, it will be 1 week that I'm gone. After that, this Zoom is not available.
Let's go.
I'm not done. I'll keep done.
And that week, if you want.